Our next speaker is Harald Mikkers. He comes to us from Leiden University Medical Center in Netherlands. And Harald is the expert in the stem cells and stem cell biology, and his talk is right as you see it on the screen, the effects of GNAO1 associated disorder variants on iPS-derived neural stem cells.
Okay, thank you, Kirill. I was just wondering whether some people have a rope here so we can tie Kirill down to his chair so he can't stand up. Because I hope I can do it within 20 minutes.
All you gotta do is stay on time.
Yes, I'll. Okay, thank you for the organization, for Stephen, Alice, and also Emily for inviting me here. It's always a pleasure to talk to such a varied audience. I think it's really great to hear all this new data, these recent advances in the GNAO1 field. And today I will discuss with you the effect of the genomic variants in neural cells. Okay, so we heard just from Terry as well that GNAO1 signaling is very complex signaling, and our lab has set up to identify the new disease or the mechanisms that underlie the disease. And since this is such a complex mechanism, I think it's a very big feat, you know, like it's going to be difficult, but we will get there. Eventually we want to get to the therapeutics stage, and this is also confounded by the heterogeneity, as you heard about. And this first we set out to investigate the heterogeneity in the global population of the GNAO1 patients. And this was published last year, and it has been flying a little bit under the radar because we had problems with human mutation and getting it indexed for PubMed. So I'm glad that Juan Dario already mentioned it here. So we did do a global inventory of all GNAO1 patients, and we came up with 282 reported patients. And here you see the heterogeneity in the symptoms, and more than 80%, as you have heard, of the patients have some movement disorder symptom, and more than 50% suffer from epilepsy, about which 60% of those have early onset epilepsy, as you've heard today as well. So if we look at all pathogenic or likely pathogenic variants in the G-alpha-O protein, you see that they are quite dispersed over the protein, except there's one desert in the middle, and that's the helical domain. So it's not the enzymatic domain, the GTPase domain. That's always, I should say, that the GTPase domain is always affected and not a helical domain in GNAO1 patients. And also you see a few hotspots there, which we have known, and those are important regions for the protein, like the P-loop and the switch regions, which are important for conversion from the GDP to the GTP situation. If we zoom further in on these hotspot regions, we see also there still heterogeneity. So some patients have only movement disorder, while others suffer from epilepsy and movement disorder, and while they are bearing the same genetic mutation. So we think that genetic background is really important. So this was published last year, and we try to keep up with all the variants that are published in the world. So right now, we have identified at least 408 small-scale genomic variants, and that doesn't mean that they're all pathogenic. So there are many benign variants, but in general, there are 408 small-scale variants in the GNAO1 gene. 116 variants are pathogenic or likely pathogenic, and so far we have in our database 309 patients identified. And all the genomic variants are deposited to the Leiden Open Variant Genome Database. So if you want to have a look at the different variants, you can have a look there. What came out from our inventory is that everybody talks about GNAO1 as one single gene, but actually the gene contains two isoforms, so GNAO1A and B. And these are just, let's see what we can do. So only the last two exons differ for both isoforms. And when you then look at the translated protein, they are very similar. They are exactly, they have exactly the same length, and they only differ in 20 amino acids. So we wondered, what is now the pathogenic variant? If we look at the number of variants found in these two discriminating exons, the GNAO1A two exons have 70 variants, whereas the GNAO1B variant exons have 132 exons. In contrast, we found 90 patients which have GNAO1 associated symptoms. 84 of these patients have the GNAO1A variant, whereas 6 patients have the GNAO1B variant. So suggesting that GNAO1A is more pathogenic than GNAO1B. If we look further into these 6 patients, 3 can be ruled out as GNAO1 as the cause, because the variants were inherited from the father or mother. So we think that GNAO1A is the predominant cause of the GNAO1 associated disorder. So we want to study the heterogeneity and the disease mechanisms underlying this disorder. So then you have to look into the genetic background, because we show that the genetic background is very important. So what you do there is you try to get cells from the patient, somatic cells, and you reprogram them into induced pluripotent stem cells, and then you want to differentiate them into the affected population of cells, in this case the neurons. Then you have your GNAO1A model, and which you can test for to study the disease mechanisms and also to see whether therapeutics work. So we start out with some variants which were found in the Netherlands, and so these are the variants. The blue ones are the movement disorder variants, whereas the black ones are the DEE variants. And then recently we acquired some extra lines via the Bo Foundation and also from Israel. Importantly, because there's a genetic difference between these patients, you need to have the perfect control. And the perfect control in this case is to repair the mutation that is found in the patient. So basically, we correct the mutation and make the cells wild type again. So what we do then is to differentiate these induced pluripotent stem cells towards neuroprogenitor cells, which are as an intermediate between the iPSC cell and the neurons. And then we want to study these neuroprogenitor cells. So first we want to see whether there were difference in transcriptomic profiles in these cells. But first you have to assess whether the purity of the cells is good, because you look at the whole population. So we did do PAC6 staining, which identifies the neuroprogenitor cells, and our criteria was like more than 90% of the cells should be PAC6 positive. So then we sequenced the whole transcriptome in these neuroprogenitor cells, and we had 40 million reads using Illumina high-sec sequencing. And then we did do a principal component analysis, which shows the variation. And this already shows you there's very little variation. So on the left we have experiment 2, on the right experiment 1. And here you see the numbers, I hope it's visible. All the genes in principal component on the x-axis only give you 0.8% of the variance. So there's more variation between the experiments than between the groups, and the controls and the mutant. So on the green, we have one pair of one set, and I should say this is a monozygotic twin set, where the isogenics cluster pretty closely, and here also there's isogenic control with the mutations that we introduced in this line. But there are some differences. So we compared all the movement disorder transcriptomics profiles to the DE transcriptomic profiles, and we didn't find any difference there. However, if we do it only for DE versus the controls, and then we do a pairwise comparison, we find 26 differentially expressed genes. There are a couple of genes, and then we did do an ingenuity pathway analysis, and what we find is that the glutamate receptor signaling, opioid receptor signaling, exon guidance, which have been also linked to G-alpha-O signaling, are affected, and also the Rho-GTPA signaling, which involves the actin cytoskeleton. So, although we have just a few outliers, it all makes sense. If we then do disorder classification, this is what we get. Everything is linked to seizure disorder and epilepsy, making sense. However, we are not interested in neuroprogenitor cells per se, because eventually the disease-causing cell is the neuron. So next we set out to differentiate these neuroprogenitor cells towards the cortical neurons, so that's the outer cells in your brain. And then we want to measure what's going on in these neurons, in these GNAO patient-derived neurons, using electrophysiology. And for that we use multi-electrode arrays, where you have basically an electrode and you glue your neurons on top of the electrode, and then when a neuron fires, you can measure the activity of the neurons. So you measure the activity, so the functionality, and also the excitability. So how often does a neuron fire? So we've done this for the E variants, and what we see here is that there's basically spike frequency, so that's activity is very similar. The burst rate, so how often that you find multiple spikes within a short window of time, is also very similar. But we do find a difference in the burst duration. So once a neuron fires, the epileptic neurons, they fire shorter. So the bursts are shorter. And then we find that the spike frequency within a burst is higher. So they fire faster, but they stop earlier. That's basically what we find. So this is for the DE neurons. So what about movement disorder neurons? We look here at cortical neurons, so we didn't expect actually to find anything. But what we do find is that motor neurons, cortical neurons derived from these movement disorder patients, they have also alterations in their neurons, and that is that the spike frequency goes up. Burst rate is higher, and also the burst duration is higher, while the spiking frequency, so the activity per burst, is lower. So to summarize this is that we find differences between movement disorder neurons and DE neurons, and that movement disorder neurons have a longer burst, whereas the DE have shorter bursts, and the spike frequency per burst is lower in the movement disorder, whereas it's higher in the epileptic encephalopathy. So, and this is good news, because now we have basically a screening system to which to discriminate movement disorder defects from epileptic defects. So we were very excited about that, and currently we are testing the other variants to see whether we can reproduce this. Just to switch gears, so basically what people have been studying as well is the localization of the G-alpha-O proteins, and for that they incorporate the M-scarlet protein, so that's what we do, into the G-alpha-O protein to visualize it under a microscope. So we've done this for the different variants, and what we see here, and I talked about this last time, is that the G-alpha-O wild type is only located in the membrane, maybe a little bit in the cytoplasm as well, whereas the two DE variants, which are on the right, they are poorly expressed and you don't find anything on the membrane. If you co-express this with the wild-type proteins, you see that the wild-type in green is in the membrane for the mutants, but for the mutants, the DE mutants, it's not going to the membrane either. This is very nice. At least we were wondering why this is, and we wondered whether this is due to the overexpression system, and also this is in tumor lines. So you use HeLa lines or neuroblastoma lines, which is a superphysiological situation. Is that truly also what happens in the patient? So then we said to ourselves, okay, we don't want to overexpress these proteins. We just want to target this scarlet protein, this red fluorescent protein, into one of the copies of the GNAO1 gene. And we use again this 2 to 9 mutant, which is a DEA-associated mutant. So we have different copies. Every cell has 2 copies. So we have 2 lines, one line where the red fluorescence is targeted to the mutant allele, indicated by the asterisk here, and the other line where the scarlet is introduced into the wild-type allele. So then we want to check what is the effect of the mutation on the localization in the endogenous situation. What we do find is that, again, as we've shown by the overexpression system, that in neural stem cells, the protein, the mutant protein, is lowly expressed in comparison to the wild-type protein. So this is where the wild-type allele was targeted, and this is where the mutant allele was targeted. But then what about the localization? What we find here, and I hope it's visible, is that the neural stem cells, we see the wild-type located to the membrane, but in contrast to the overexpression experiments, also the mutant is located to the membrane, which is contradictory to what we found in the tumor cell lines. So then we differentiated these cells, and this is really recent data which I wanted to show you, towards neurons and see what's happening in those neurons. And this is what happens in the neurons. On the left we have the wild-type allele targeted. We see pure membrane localization with some aggregates formation, but in the mutants there's less membrane localization and there's more aggregate formation, indicating there are differences there. So now the next question is, what's the localization in neurons? Is this to the Golgi? Are they located to the lysosomes? What's going on there? But also this gives us a new handle, a new drug screening tool, because now we can test drugs and look at the localization of the mutant protein, and also see at the effect of on the expression levels. However, you need to next is also to test the effect of the mutant protein. So is there a dominant negative effect? We want to study that, and for that we also generated. So basically the 2 lines that we have, we repaired the mutant allele, or we made a homozygous mutant. So now we have the full set of targeted lines, iPS lines, which we can use to study the different questions that we have. This is a summary, but I think due to the time, and I don't want to stand up here, I'm going to skip that. And these are the people that contributed in blue, the people from Leiden University, and I really have to thank the people that made this research possible. So that's the Dutch Foundation, GNAO1 Foundation, the For Wisdom Foundation, and also the Bow Foundation. Thank you for your attention.
Thank you, Harald, for this very exciting findings. We've got time for questions.
Thank you very much for a very interesting talk. I was curious, or I found it interesting, the distinction between the epilepsy and the movement disorder patients and the cortical neurons. And I wondered if you'd studied medium spiny neurons, and if you'd expect to find maybe something opposite, just because presumably the cortical neurons are more involved with epilepsy, whereas the medium spiny might be more responsible for the movement.
Yeah. These experiments are they are really time consuming. So we've been thinking of making spiny neurons, and then we skipped out from it. So, and especially now also, since we find a phenotype for the movement disorder, we want first to validate whether this is true and see how we can use that. Because what we know from other studies is that the effects that's seen in the patient are normally exaggerated in a dish. So it might be that the cortical neurons in a patient are normal, but that we find symptoms in a dish because they are out of their comfort zone. Yeah.
Hi. So we, together with Simone here and Alessandro Rosa from Italy, did the iPS-derived neuronal analysis using two independent patient-derived fibroblast lines having the mutations in the G203R position. And one phenotype which is consistently coming up is the defect in the neural rosette formation. I wonder whether this is specific to the G203R or whether you see it also with the mutations you have studied.
We think we see a difference there, although it's not as obvious as you guys have published. We think there's a difference in polarity. So we did do ZO staining. So ZO1 shows you the polarity of the neural rosette, which is kind of similar. We need to do quantitative analysis on that, but we think there are differences, and especially we think that the rosettes from the mutants, from all the different mutants, are more monolayer, and that the rosettes from the isogenic controls, they're more 3D formed. So we think there's a defect as well. Only right now we are more interested in the neurons, and we are not going to pursue that any further. Yes.
There's a question there.
Yeah.
In alternating hemiplegia of childhood, which is another disorder, which with dystonia and epilepsy in childhood, there's really beautiful evidence from Catherine Svoboda that there's accumulation and aggregation of the disease protein. It's kind of like those punctate spots. Could be very different, but you had a chance to look for beta subunits and gammas to see if they're getting properly handled in the Golgi and the ER and these beautifully differentiated cells.
Yes, no. You're absolutely right. And we think that those are indeed aggregates. We looked at Golgi, but it's not, we don't see a pure overlap with the Golgi. So it's more like cytoplasmic, and they are retained in cytoplasmic vesicles, aggregates. We haven't identified the organelle structures or what they are in, and that's our next task to do so. But indeed, the 229 and the 278, so the DE-associated variants, they are, according to Dolman, classified as group 4 variants, which are very unstable variants. They seem to form aggregates, and very similar aggregates. Yeah, and we don't know what's what, because also the next question then is like, how do these interfere with regular GLFO signaling, right? Because they don't get to the membrane. So is this indeed a neomorph, or is there something else to it? And to be honest, we don't know yet.
Well, I have a quick question. So when you look at the list of the differential expressed genes, you have at least 2 different mutant lines.
Yep.
Do these changes seem to be different, or are they the same that you pick up? I might have missed that.
So now we find 26 common outliers. So that's a 229. So those are 3 lines. So one from a monozygotic twin, that's a 278 line, and one also the 229 line, which are classified as group 4. So those are the unstable variants, which give early onset epilepsy. And then we find 26 genes deregulated, and that's what I showed you. They are all linked to epilepsy, right? And yeah, we haven't analyzed all the genes yet. Yeah.
I think we
No
should
more
leave it
questions?
there. There's a lot of questions.
All
but
right.
I will sense of
I will
fact
be
we're-
in the there for lunch.
Yeah,
so
great.
Then you can ask questions, right?
Speak